Hello, welcome to our own channel, Curious Corners in Biology, where we discuss scientific aspects of this subject and things about biology which makes this subject worth curiosity, worth scientific curiosity, I would say. So today, as usual, we are going to discuss couple of questions. So often we have heard that certain pathogens, say for example, causing tuberculosis, pneumonia, certain bacteria have the capability to evade the immune system of the body. Had it not been so, then they would have been cleared before even infection sets in. Okay. But infection does set in and microbes are not easily cleared off. It requires treatment, it requires some amount of exposure to antibiotics, at least if not prolonged. So, one of the substances which I would like to discuss today is wax D, that is responsible for helping microorganisms evade the immune response. Okay. Now we have all heard about common wax. Wax chemically is ester of long chain fatty acids and alcohol. Okay. We are not talking about normal wax. We are talking a chemical substance which is expressed on the cell membrane of the pathogen, and that is wax D. Wax D chemically is LAM, which is lipoarabinomannan. So that means it is a complex of lipids and sugar. In simple words, it also contains sulfatides and glycolipids. Okay, so it is a complex molecule. You can consider it a conjugated lipid sort of molecule, lipid coupled with sugar. Okay, especially the sulfatides and the glycolipids in wax D will help in the mechanism of evading immune response. Now, how does bacteria like, say, for example, Mycobacterium leprae, Microbacterium tuberculosis, how do they escape the immune system of the host? So, what happens is usually macrophages, which are the natural immune cells of the host, will be sensitized and they would march towards killing of the pathogen. But here is the catch. Macrophages will be even able to engulf the pathogen. So phagosome is formed. Okay. Usually when a cell capable of phagocytosis is engulfing anything, we call it phagosome. But then phagosome, due to presence of wax D, is not able to interact or integrate with lysosome. So if it is not able to integrate with lysosome, that means this pathogen cannot be destroyed by the acidic pool of the enzymes of lysosome. So the pathogen remains as it is, even though phagosome is formed. Are you getting what I'm saying? So we can understand this by drawing a small schematic diagram as well. You will be able to see my screen. Let us say that this is the macrophage I'm talking about. Let's say this is macrophage. Okay. So this is a macrophage. This is a large cell, irregularly shaped cell having nucleus and cytoplasm. Okay. And let's say here we have our beautiful pathogen. Okay. Now this is a pathogen. Okay. So usually what macrophage does is that this is going to engulf the pathogen. Okay. So it is going to form pseudopodia like this. Okay. And eventually it is going to engulf this pathogen. This pathogen gets in. Okay. So can I say that something like this can be formed? Now this pathogen, of course, after phagocytosis is covered by a membrane. Okay. So here is the pathogen and a portion of cell membrane is given off to encircle or form a boundary around the pathogen. So this is called as phagosome. This is what is called as phagosome. Now, is the story over now? No. This phagosome also has to be destroyed. So, of course, any macrophage would be high on lysosomes. Let's say that these green colored structures are lysosomes. Okay. So, the lysosome is supposed to act on the phagosome. But if lysosome is not able to interact or act on, if it is not able to act on phagosome, then the pathogen remains intact. And this is how the immune system of the host is evaded. That means it is escaped by the pathogen and pathogen manages itself to stay put, causing continual infection in the cells of the host. Okay. So this is wax D, and this is one of the ways how pathogen can evade the immune system of the host. The next question I would like to discuss today is that we know that DNA is a genetic material. Okay, that's a Bible truth. Everybody knows. But there are forms of DNA as well. There is A DNA, there is B DNA, there is C DNA, and nowadays pDNA, eDNA, even two more forms of four-stranded DNA have been discovered in cells, and that is I motifs and G4 quadruplexes. So this goes on and on. Okay. But we know that the most common form of DNA existing in, say, vertebrate cells or human cells is bDNA. Now, how do we differentiate different forms of DNA? It's very simple. They are differentiated on the basis of parameters like diameter of DNA, what is the distance between base pairs, what is the rotation of the DNA, right-handed or left-handed. So bDNA is the most common form of DNA. Okay, B for Bombay. Then the other three common forms of DNA, or other two at least, would be aDNA and zDNA. cDNA is also more common, but as far as I know, that cDNA is mostly synthesized in laboratory. Okay. So let us consider aDNA, zDNA, and bDNA to be existing. So one question which can arise is, are these forms of DNA interchangeable? And if they are, do they interchange? And if they interchange, then what is the most abundant form of DNA if they do interchange? Okay. So, yes, there are answers to this. And yes, interchanging of DNA is seen in human cells as well. No doubt, bDNA is most prevalent, most common form of DNA, but this bDNA, depending on ionic environment in the cell, level of hydration in the cell, or you can say level of salt content in the cell, these are the factors which influence the interconversion or interchange of the forms of DNA. So usually it has been found out that aDNA form is favored by low hydration condition. That means when cell has low water potential or is scarce in water. Okay. Then zDNA is favored when cell has high salt concentration. Okay. So high solute concentration in the form of salt as a solute can favor zDNA formation. Okay. Then one of the things to remember is that aDNA is favored by certain specific stretches of purines and pyrimidines along its specific length. So say for example, there is a specific length in aDNA, then that length of nucleotide should be either occupied by purines or pyrimidines. Purines and pyrimidines cannot alternate with each other. And zDNA, on the contrary, would show purines and pyrimidines alternating as nucleotides along a stretch of DNA. Okay. So the base sequence, or you can say the nucleotide sequence, also determines the form of DNA which is prevalent in a cell, or if the nucleotide sequence is changed, there is a possibility that the form of DNA is also changed. Okay. Then there are certain proteins which can bind to DNA and change its conformation. Because if the conformation of the DNA is changed, then the form of the DNA is also changed. Okay. So how interesting. It's really fascinating that DNA is not just single typical stereotypical molecule. No, 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 no. It also has diversity. Diversity is ingrained in the genetic material itself. Diversity is not just on the level of individuals, it is at the level of DNA as well. Okay, it is at the level of DNA as well. And of course, I motifs and G4 quadruplexes, which I talked about, have been known to be cytosine rich and have been researched recently to be present in locations like telomeres, which are the ends of the chromosomes. Okay. We just know double stranded DNA, but there are four stranded DNA as well. Okay. Then, another interesting question is, if something is living, do we consider it to be in equilibrium or in non equilibrium? Okay. So, for those of you who are biologists, would know the answer to this. Equilibrium is where no work is done. Okay. The rate of forward reaction is equal to the rate of reverse reaction in equilibrium. But if this prevails, then a living system would not even survive, because death is equilibrium. Death is equilibrium. There is no change, there is no work, there is no reaction, there is no metabolic flux. So obviously living state should not be in equilibrium. A system, if it is living, has to be in non-equilibrium. Only a state except equilibrium state can ensure metabolism, and metabolism is characteristic of life. Okay. Then the next question I want to discuss is, we have heard that many bacteria, say for example Thermus aquaticus, Taq polymerase is the name of the enzyme derived which we use in PCR, polymerase chain reaction, that has been derived from the bacterium Thermus aquaticus. Then there is um, Bacillus terothermophilus. Okay. The characteristic of these bacteria is that they are thermostable. That means they can survive at a temperature higher than 50 degrees Celsius or even more than 80 degrees Celsius. They can thrive. So, what is the reason for that? The answer to this is they have well equipped or specialized proteins and enzymes which will allow them to perform metabolism under such hostile temperature conditions. Okay, accepted. But then, not all bacteria, not any life is capable of surviving in extreme conditions or hostile environment. Usually, living organisms survive below 50 degrees Celsius, or so to say, below 40 degrees Celsius. Then, if thermostability is at the level of proteins or enzymes, then what is that in proteins or enzymes which confers thermostability? Yes, we know that enzymes should be thermostable for any kind of metabolism to occur or to go through. Yes, correct. And enzymes are proteins. So definitely there should be something at the level of enzymes and proteins which confers thermostability, because enzymes are and proteins are the functioning biomolecules of the cell, the whole function of the cell. The regulation of the functioning of the cell is in the hands of enzymes and proteins. Okay. So the reins are in the hands of enzymes and proteins. So what makes them thermostable? So there are a couple of things which contribute to the thermostability of enzymes and proteins. And those are bonds. The more hydrophobic bonds occur in protein, the more stable the protein would be. Because hydrophobicity of the protein would make protein more compact. And anything which is more compact, remember, it cannot be linearized easily. And if it cannot be linearized easily, it cannot be denatured. And if it cannot be denatured, it cannot be made dysfunctional. The native structure of protein, when acting as enzymes or any functional protein, is globular. So it is compact, it is spherical, it is compact. So heat can denature it. But if there is something in the protein which will help to resist the denaturation, then the protein will be able to hold itself up into its functional structure, that is globular structure. One of the things which contributes to it is hydrophobicity. Then there are different electrostatic interactions within the protein molecule itself, like you have salt bridges, you have disulfide bonds as well. Disulfide bonds will stabilize the protein as they are very strong covalent interactions. So more stronger the bond, more energy would be required to break it. And of course, bonds need to be broken in order to denature or linearize the protein. So of course, disulfide bridges, salt bridges, electrostatic interaction, that means ionic bonds as well. Okay. Then there is another factor which is called as cyclization enzymes. These are specialized enzymes which will join the N terminus, that is the amino terminus of the protein, with its C terminus, that is the carboxyl group of the protein. So when two ends are joined, the protein is held up as a globular structure. Okay. So again, we arrive at a globular structure, the compactness and the fitment of the protein will be more towards a functional significance. Okay. The protein will be made globular. So cyclization simply means converting any linear structure into globular structure. All that we want is globular protein for it to function and for it to be stable. Another factor which is contributing to thermostability is also glycosylation. Glycosylation is addition of carbohydrate moiety into protein. And glycosylation has also been known again to increase the rigidity of the protein. Now, if the rigidity is increased, then the structure becomes stable. It doesn't become more loose, it will become more organized. So, in this way, protein will not be denatured as well. Okay. So these are different bonds, or you can say different interactions present within the protein, which gives a characteristic of thermostability. Okay. Now, these factors are not usually present at the level to which they are present in thermostable protein in normal proteins. In thermostable proteins, these characters which I talked about, these factors are more pronounced. That is what I mean. So, hope you understood. The next question I would like to discuss is, we know that enzymes lower the activation energy, that is the barrier which has to be crossed by the reactant to get converted into product. Okay. Do ribozymes lower activation energy in order to catalyze a reaction? Yes, of course. They work on the same principle. One of very good example of ribozyme is peptidyl transferase. Ribozymes are usually isolated from plant viruses as well. Okay. So yes, ribozymes also do have this potential of lowering activation energy and functioning usually just as how enzymes do. Now, one of the interesting things about ribozyme or any enzyme which has been researched is promiscuity of the enzyme. Now, this promiscuous word feels a little bit, what do you mean by promiscuity? Promiscuity simply means over here non-specificity. Okay. The enzyme has a degree of non-specificity to its biological property, even though we know that enzymes are specific for substrates they act upon. But for any enzyme, it has been found out that it does have a capability to interact with certain other spectrum of substrates which it usually does not in the metabolism, and same applies for ribozyme as well. So enzymes evolved in a way that they took up the substrates which they considered to be specific, and depending on the product requirement they might have been selective for the substrate. But the inherent property of the enzyme is, yes, that they are promiscuous. That means they are non-specific. They can go with other substrates as well to catalyze a particular reaction. Okay. And the final question which I would like to discuss is, we know that when someone cuts onion besides us, then we have teary eyes. Okay. Tears just start to flow from our eyes. Why so? What is happening? What magic is happening? Okay. So this is actually very simple. When onion is cut, remember the cells of the onion are also cut. So cells of the onion would release a gaseous substance called as amino acid sulfoxide. Now this amino acid sulfoxide will irritate our lacrimal glands. Of course, there are intermediate chemical reactions like amino acid sulfoxide gets first converted into sulfamic acid, and then it is getting converted into one more intermediate, and that is a sort of oxide of propane. I don't exactly remember the name, but sort of a propane oxide. That's a volatile gas, and that irritates the lacrimal glands. And do you know why lacrimal gland upon irritation secretes tears? These tears are used to flush out that oxides which have entered our eyes. Okay, the sulfoxides or the oxides of sulfur which have entered, which are irritating, they are to be flushed out through the modality of tears. Okay, that is why lacrimal gland is sensitized. So, hope you enjoyed the discussion. Okay, we will be, of course, coming up with many, many, many such questions. If you are interested in such content, please visit the link in the description. I have written a book, Curious Corners in Biology, which is a repository of such curious questions in biology. That's available at a very affordable price. Okay, you can buy the book, and you will definitely arouse curiosity in your mind, and this will help you in your cognitive progress, I would say. Okay. So that much for today. Thank you very much. See you.